Good afternoon, everyone. I'm Cliff Brangman. I'm the director of the Omenn-Darling Bioengineering Institute, and it is my pleasure to welcome you to the third annual Gilbert S. Omenn, MD, 61, Martha A. Darling, 70 lecture in ethics and policy in bioengineering. This is a special event for us, and I'm glad you're all here with us this afternoon. We began this lecture, the bioethics lecture, in the 2022-2023 academic year as a signature event of what at the time was called the Princeton Bioengineering Initiative, whose goal was to support and expand bioengineering activities already underway on campus and ignite new directions in research, education, and innovation at the intersection of life sciences and engineering. And as I think most of you know, last summer, it was actually summer of 2023, and then last year we had the official launch party. We launched the new Bioengineering Institute, which is called the Omenn-Darling Bioengineering Institute, which was enabled by the tremendous generosity of Gil Omenn and Martha Darling. And we're very glad to have Gil and Martha here for today's lecture. So let's take a moment and thank them for their support. So Gil and Martha recognize the transformative impact of bioengineering for society. And we're seeing this every day, from the development of mRNA vaccines, antiviral therapies, to CRISPR gene editing and CAR-T-based cell engineering for immuno-oncology. The impacts are really everywhere, and we're seeing them every day. But as with other powerful new technologies, there arise a host of ethical questions, and we think it's critical to stimulate conversations and build bridges across campus, including with our colleagues working in the humanities, policy, and social sciences, for example. So we're grateful to Gil and Martha, not only for their strong support of Princeton Bioengineering, but also for helping to facilitate and stimulate connections with these different units on campus to engage us all in this really important conversation. So thank you. This conversation has been spreading organically on campus. This includes the formation of a student-led group called the Princeton Bioethics Exchange that grew out of our first Omenn-Darling Bioethics lecture. We'd also like to thank the School of Public and International Affairs, the University Center for Human Values, the Faith and Work Initiative, and the Council on Science and Technology for their partnership and support of these events over the last several years. It really has been wonderful to see all these units coming together and to stimulate this discussion. So I'm very pleased to announce our 2025 Omenn-Darling Bioethics lecturer, Hank Greely. Hank is the Deane F. and Kate Edelman Johnson Professor of Law at Stanford University and Director of the Center for Law and the Biosciences. He's also a professor by courtesy of genetics at Stanford School of Medicine and chair of the steering committee of the Stanford Center for Biomedical Ethics. Professor Greely is one of the foremost thinkers on the ethical, legal, and societal implications of emerging technologies, emerging biotechnologies. He's written extensively about the implications of new technologies with a focus on neuroscience, genetics, and human reproduction. He's written several books, including The End of Sex and the Future of Human Reproduction. Provocative title.
Available at bookstores everywhere. As long as they're called Amazon.
And CRISPR People, the Science and Ethics of Editing Humans. So, Hank, we're absolutely thrilled to have you as this year's Omenn-Darling lecturer and excited to hear your insights on the bioethical dimensions of transformative developments in bioengineering. Thank you.
Great. Well, thank you so much. I am equally pleased to be here. I realized yesterday, suddenly I woke up and said, Tuesday, March 4th, it's Fat Tuesday. This is Mardi Gras. Now, I know that at an institution like Princeton, founded by undoubtedly dour Presbyterians who would have viewed Carnival as papist nonsense, it may be a little wrong for you to start by invoking Mardi Gras. But I lived for a year in New Orleans, a wonderful year in New Orleans. My liver, I think, lived 3 years in the year I lived in New Orleans. But I want to start with some music. So here we go, I hope.
Medicine is magical and magical is art. The boy in the bubble and the baby with the babbling heart.
So I noticed some people nodding, and I noticed a number of people who weren't born when that song came out. That's 1986. The great Paul Simon on his wonderful Graceland album. The song's called The Boy in the Bubble. And I love the lyrics. I love the idea of these are the days of miracles and wonder. This is don't cry, baby, don't cry. And he calls out medicine is magical and magical is art. Think of the boy in the bubble and the baby with the baboon heart. And that's what I want to do. I want to walk you through some of the issues and some of the reasons to take issues seriously that are ethical, legal, social, political, cultural issues raised by biotechnology in all its various guises. Specifically, I want to do three things. First, I want to talk about some of those miracles and wonders, which are also sometimes terrors and deep fears. Second, I want to talk about why it's been more complicated than we expected. And third, I want to talk about Frankenstein and the lessons of the Frankenstein novel for our current and future lives with biotechnology. I'm going to do it without slides. I've never been sure whether I am allergic and it causes an anaphylactic shock, or whether it's against my religion and would damn me to eternal hellfire, but I don't do slides. To be honest, it's because A, I'm old and I didn't want to learn, and B, I've never been to a conference where somebody's slides didn't mess up. And so I figure if I never use slides, they can't mess up on me. So first, miracles and wonders. Let's talk about the boy in the bubble and the baby with the baboon heart. These were real things. The boy in the bubble was a boy named David Vetter. He was born in Houston, Texas. He had a very serious problem called severe combined immune deficiency. He lived for 12 years in a bubble, pretty much in a bubble. There were times when he could be out a little bit. He rarely was able to touch any human. And when he did touch people, it was through gloves on both sides. There was a made-for-TV movie about him. He had to be in that bubble because he had no immune system. And with no immune system, it doesn't take much of a pathogen to kill you. Eventually, when he was 12, he died. There are now, I think, known to be at least 16 different forms of SCID, severe combined immune deficiency. Only 2 or 3 of them are uncommon, and the rest are vanishingly rare, including one that was discovered by a 4th-year Harvard medical student many years ago. I was told there'd be no feedback, come on. It's thought to be found in about 1 in a million births, so maybe 3 times a year in the United States, 3 to 4 times a year in the United States. It's called Omenn syndrome, and I forget who discovered it. So SCID still happens, but now, unlike then, we have some interventions that are better than putting a boy in a bubble. We have some interventions ranging from drugs to stem cell transplants, bone marrow transplants, to gene therapy. They're not perfect, but they're a lot better. Miracles and wonders. The baby with the baboon heart was a baby named Faye. She was born in Southern California in the Inland Empire, east of LA. She had a serious heart defect. It was so serious that she needed a heart transplant. She needed it quickly. This is 1983, I think, and heart transplants were a thing, but baby heart transplants were trickier, and they didn't have a heart for her. She was treated at Loma Linda University, which didn't have a heart for her, but had 9 baboons. And so they sacrificed a baboon and put the baboon's heart in baby Faye. She lived for 21 days with the baboon heart and then died. It was controversial at the time, and it remains controversial about whether that was an appropriate kind of intervention. But today, 6 different living people have received pig organs. 3 have gotten kidneys, and one of the ones who got a kidney just went home from the hospital recently, within the last week. 2 got hearts. They didn't survive. They survived for 40 or 50 days. One in China got a liver, and I can't find out what happened to that patient. It was like last summer, and I can't find any news, but they said he was fine. What's happened? Well, we've gotten better at immune suppression, but we've also gotten better at genetically modifying pigs to make them, to take away the markers that cause the human immune system to go, well, bat shit, pig shit, ape shit, to go crazy when you get a foreign species organ in them. Interestingly, some of the first of these transplants were done not into living humans, but into the living bodies of dead humans. People who were declared brain dead had kidneys transplanted into them or attached to them, which I think raises another set of interesting questions, ethical questions about using the dead in that way. But again, miracles and wonders. FDA has now approved a clinical trial. The cases in the US that have been done under something called compassionate use. But there's now a clinical trial, and there are 2 different companies modifying pigs to be used this way, because there are tens of thousands of Americans who die every year waiting for an organ. And there are a lot more pigs than there are human organs that are available and healthy and good for transplant. So Paul Simon, 1986, both of his examples have changed, miracles and wonders, but neither one is, in full, perfect clinical use yet. I want to now spend a few minutes running through, in really quick fashion, a bunch of other strange things on the edges of biomedicine, dealing with tissues, cells, and organs, dealing with brains, and dealing with genetics. So let me start with some work in human neural organoids, much of it done by my Stanford colleague, Sergiu Pașca. These are little balls of human brain cells. They're about the size of a pea, but even though they're about the size of the pea, they can hold 1 or 2 million neurons. And some of them have lived for 3 years, and they keep changing and making new connections, and some of the neurons keep changing from one kind of neuron to another kind of neuron. We don't quite know what's going to happen with them ultimately. 1 to 2 million neurons is not a lot. All of you have roughly 89 billion neurons in your brain, but a honeybee has 1 million neurons. And honeybee is capable of some pretty sophisticated behavior. Sergiu has run some of these things together. There's a limit on how big they can get because they don't have blood vessels. And so they form spheres, and the interior of the sphere gets cut off from oxygen and sugar, and it dies. So they're hollow spheres. People are working on vascularizing them, but they haven't yet. They will. Right now, they can't get very big, but Sergiu has strung them together like pearls on a string and connected them into what he calls assembloids. He has a 3-part assembloid. One part receives information, one part made of neurons from the cerebrum processes it, and the third part are neurons that are motor neurons that then are attached to a piece of muscle. He stimulates the first part, and the muscle twitches. What are those? They're not animals. They're not humans. What are they? What can we do with them? What should we do with them? One thing Sergiu has gone farther with is transplanting these into baby rats. He published a cover story, if cover story is the right word, in Nature in late 2023. He had moved these into a bunch of newborn rats. The human organoids grew. The rat vasculature, the rat blood vessels, fed the human organoids. He ended up with rats, 1/6 of whose cerebrums were human organoids. He then did some cool experiments. He tickled the rat's whiskers, and he saw that the human cells fired. So not only was it being fed by the rat vasculature, it had synapsed with, it had made connections with the rat brain. Then, using my Stanford colleague Carl Dazeroff's method of optogenetics, he stimulated particular areas of the human organoid, and when he stimulated them, the rats went and drank, and when he turned the light off, they stopped drinking. So he had connections both in and out between the rat part of the brain and the human part of the brain. What is that? Happily, it didn't stand up on its hind legs and say, "Hi, I'm itchy," or "scratchy," or whichever was which. I never figured that out in The Simpsons. But it's odd. Second suggestion, embryo models. Embryo research human embryo research has been quite controversial ever since we were able to get our hands on human embryos. Interesting history, it goes back over 100 years with early embryo work, but the big deal came after IVF made embryos available for researchers to do research. And since the early 1980s, there has been this both formal and informal 14-day limit on how long you can do research with embryos. Well, these are embryos that are made from an egg and a sperm. They're usually left over from IVF. But researchers have been making embryos with induced pluripotent stem cells, or human embryonic stem cells, putting them together, no egg or sperm directly involved, and letting them develop. Some of them have all the equipment they need to be an embryo. They've got the extra embryonic tissues, for those of you who care about such things, yolk sacs and so on. Some of them don't and are only partial. The ones that are only partial, there's probably no way they could develop into a baby. But the ones that are fully integrated, we don't know what they could be. Do they count as embryos for the 14-day rule? Legally? Ethically? Should we think of them as embryos? There is, of course, an experiment that I think could resolve at least this in one direction if it went one way. If you put them into a woman's uterus and they turned into babies, I think we'd have to say those are embryos. But we can't do that experiment. There are lots of ethical problems with doing that experiment, mainly that it would be incredibly risky to whatever it is that might come out at the end of those 9 months of gestation. So what do we do with embryo models is something that's been kicked around a long time recently. Newer issue. Life is handed. Kind of actually, in this case, I think it is purely binary. Molecules are either left-handed or right-handed, which doesn't actually have to do with anything you can see. It's the direction in which light skews, polarized light skews when it goes through a solution of them. Proteins are left-handed. Gil, right? DNA, nucleic acids are right-handed, proteins are left-handed, I believe. Okay. One or the other. People have been making things with the wrong-handedness. Backwards proteins, mirror life. So look at yourself in a mirror. You think that this little zit is on this side, but actually it's on that side because the mirror image is different. The right-handed amino acid, the right-handed proteins are different from the left-handed proteins, and all life on Earth uses right-handed proteins. But maybe not for long, because people have been trying to figure out, can they make left-handed proteins? Can they make life from left-handed proteins, and what would that mean? Just about 4 months ago, a group of scientists doing this got together and said, wait a second, if we had a bacterium that had the wrong-handedness, would anybody's immune system be able to recognize it? Could it kill everything? And so currently there is a scientific recommendation to have a moratorium on this unless or until we can ever conclude that it would be safe. It is changing life around. Why would you want to do it? To see if it works, to see if it makes a difference, why it makes a difference, maybe to try to figure out why life only has right-handed proteins. We don't know, but it's dangerous. Last one in this category, in vitro gametogenesis, which is a long-winded term for making eggs and sperm in a dish. We know how to make eggs and sperm. I'm willing to guess that everybody in this room has made either eggs or sperm, though probably very few, if any of you, have made both eggs and sperm. It all starts out in the embryo, and it goes into the yolk sac, and it moves around. It's amazing that anybody ever gets born, because human reproduction is truly weird and bizarre. But at some point, you've got something that is not yet an egg or a sperm, could go either way. If it's put into an ovary, it becomes an egg. If it's put into a testes, it becomes a sperm. Japanese scientists over 10 years ago did this with mice. Remember, just says in mice, important thing to remember with all scientific stories. So scientists cure Alzheimer's in mice, but with mice, they were able to take induced pluripotent stem cells, so cells that you can make turn into anything, which we can take from skin. So we could take a presidential stem cell from Dr. Tillman's skin. We could turn it into an induced pluripotent stem cell. And this has been really exciting. Shinya Yamanaka from Japan figured this out in 2007. 5 years later, he got a Nobel Prize, which is almost a world land speed record for a Nobel Prize. It's been really exciting. The hope is that if she needs heart cells or liver cells or kidney cells, we can make them, and they will be hers. So her immune system will not rebel and try to kill them, and we won't need immunotherapy. Well, eggs and sperm are cells. And I was at a meeting in 2010 where a friend of mine said, as part of a 40-minute lecture, one sentence that stuck with me. And what would happen if we could make eggs and sperm this way? Well, I thought, that's an interesting question. And 6 years later, I had managed to find the 6 months it took me to write The End of Sex and the Future of Human Reproduction, which suggests that we will be able to make eggs and sperm from people's skin, and that will be kind of revolutionary. Why would we do it? There are millions of couples who desperately want to have genetic kids. Now, do they need genetic kids? How important is it to have a genetic link? Philosophers love to say, well, it's not really a big deal. It's a desire. It's not a need. But tell that to the people who are in great suffering because they can't have kids that are genetically theirs. They would love to have this. Or, in the Japanese group, Mikanori Saito's group did this last year. They got sperm from a mouse the old-fashioned way, and I don't want to think too much about how you do that with mice. They took skin cells from a male mouse and turned them into eggs. And so they made mouse babies with two fathers. There are a lot of couples in the world who would really love to be able to do that. Gay and lesbian parents who would really like to have kids that are 50% one of them and 50% the other. This opens that possibility. But it also opens possibilities like if somebody were to throw a coffee cup away and I fished it out of the trash and I got some skin cells off of it, maybe I could make you a father 100 times over. Or maybe a mother, at least genetically. There are all sorts of possibilities. One being, the hardest part of in vitro fertilization is egg harvest. It's the most expensive. It's the only part that's dangerous and unpleasant, apart from the expensiveness. If you could harvest eggs, instead of harvesting eggs, if you could take a 2 millimeter punch biopsy of the skin and turn that into 20 eggs, 50 eggs, 100 eggs, 1,000 eggs, 1 million eggs, IVF becomes a lot easier. And people could make, say, 100 embryos and test each one through whole genome sequencing and pick the embryo they most liked. It's not a designer baby, it's still selected. If the parents don't have the alleles, you're not going to get them in the baby, but it gives them many more choices. So that's another weird thing that I think is going to happen. Let me talk about brains. First, Nanad Sestan, a neuroscientist at Yale, a couple of years ago, made a deal with a local slaughterhouse. They agreed for pigs that were about to be bacon or pork chops, they would cut the heads off of them, and instead of turning the heads into sausage, they would hang them on a hook and let them drain for 4 hours. And then Sestan's people came and collected the heads, took them back to the lab, hooked them up to a normothermic machine perfusion unit, which meant blood temperature, normal body temperature, perfusion, fluid going through. It wasn't blood, it was a acellular perfusate. It was something that didn't have cells that could clog up in capillaries, but it had glucose and oxygen and other good things. Nanad is a careful guy. He was careful to say, not to say the brains came back to life, but the cells came back to life. Conventional wisdom is after 10 minutes, neurons in the brain die. If you're lucky enough to drown to death in a really cold lake, maybe you've got 30 minutes, 4 hours. So what happens if that's a human brain instead of a pig brain, and you hook it up, and the brain comes back to life? He did see firing, he didn't see function. He didn't see patterns of firing. He couldn't get an EEG, but it also put a factor into the perfusate to repress neuronal activation because he didn't want firing. He didn't want an EEG. Because if he gets an EEG, does that mean that pig is alive? Maybe. Brain-computer interfaces. So I will say this is one of many things from Elon Musk you should not believe. Neuralink is not going to let us all communicate telepathically in the next 2 years, 20 years, probably 200 years. But brain-computer interfaces are getting better and better. Groups are doing them for quadriplegics or other people with a locked-in syndrome of one kind or another. I believe that the Stanford group, led by Jamie Henderson, set a new record, 62 words of email per minute. Now, I took typing in junior high school and summer school, and I was pretty good. I got up to like 65. Theirs aren't perfect, but you can get a typist job at 62 words a minute if you're 80 or 90% accurate. And this is with electrodes in the brain. It's not a fun and easy thing. That's why we're not going to get it done to ourselves very often, but it's working. Another one, a group in Switzerland, took somebody who had a spinal cord break, figured out a way to decode signals coming from the brain down the spinal cord, short of the break, bridge them over with an electronic bridge to below the break, and put back in below the break the messages that were supposed to be coming from the brain, and that paraplegic is walking. He's not, I think, doing gymnastics, but he's walking. This is the age of miracles and wonders. None of what I just said is yet available clinically, but it's there on the edge. And last, let me talk a little bit about genetics. Second book I wrote was CRISPR People. That was about the terribly unethical research done by He Jiankui. And those of you who speak Mandarin, please forgive the way I've undoubtedly butchered that name. Real nightmare for an author because his family name is spelled H-E. So if you're writing a sentence and you start with H, capital H-E, are you saying he or are you saying who? Difficult for an author, right? So he took couples where the husband had HIV, the wife did not. In China, those people are not allowed to have kids. They're not allowed to use IVF, they're not allowed to use assisted reproduction. China doesn't have very many HIV cases as a percentage, although out of 1.3 billion people, it's a fair number of people. And it's heavily stigmatized. And he said, give me your embryos, I'm going to have a genetic vaccine for them. And he took their embryos, paid for their IVF, took their embryos, used CRISPR, clustered regularly interspaced short palindromic repeats. I actually emailed with the two guys who named it CRISPR, and they said part of why they named it that was sort of reverse engineering, so they could get a name that wasn't being used and that was pronounceable, was pronounced easily, and turns into a verb and an adjective and all sorts of things. So he CRISPR'd embryos, which led to the birth of 3 babies. He announced this in Hong Kong at the second global global summit on human genome editing. I believe he genuinely thought he was going to be hailed as a hero. He is not a man blessed with a lot of self-awareness, I think. He was instead reviled. He left the stage in a huff, and he was not seen again in public for over 3 years, 3 years of which he supposedly spent in prison because the Chinese government convicted him of various crimes, although China is not very good about explaining anything. Those babies are now over 6 years old. We have no idea what their health is. Chinese government wants to protect their privacy. Like 3 6-year-olds in a population of 1.3 billion, you can't reveal anything? We don't know. People are worried about super babies, about X-Men and mutants and so on. We don't know how to make those. We don't know the genetic variations that lead to above normal ability. We know a lot about genetic variations that lead to below normal activity and ability. But people are going to buy into this. And even though there is, as close as I've ever seen to a consensus in the scientific community that this is a bad thing, some of that is based on we don't know whether it's safe, which is clearly true. Some of it is based on even if it's safe, we shouldn't mess around because unlike gene therapy in a living person, a baby, a boy with SCID, you give him gene therapy. You fix his stem cells. He has an immune system, but he doesn't pass that variation onto his kids because it needs to be in his sperm to pass on to his kids. But if you edit an 8-cell embryo, it should at least get into every cell, including the cells that become sperm and eggs. It is transgenerational genetic editing, and that's really controversial. So we've got that on the human side. I actually think the non-human side is going to be more important, mainly for agricultural purposes, and mainly because we're willing to risk things with calves and baby, colts and even puppies and kittens that we won't risk with human babies. We're going to do, and with plants, we're willing to risk a whole lot. We're going to do a lot of it. Some of it will make a lot of sense. Some of it will be frivolous because, I hate to point this out to you guys, but we are fundamentally a frivolous species. So you can already buy a glow-in-the-dark petunia. It's not a glow-in-black light. It's not fluorescent. It's phosphorescent. You can order it through Amazon and get a glow-in-the-dark petunia. You can get tropical fish that are fluorescent, so you need to have an ultraviolet black light. They will glow in different colors. We will do that. If you have not yet looked at today's news, you should see in today's news pictures of a couple really cute mice. Mice that have long, furry hair. These are, I don't know, the mouse that trumpeted. They're not the mammoth that squeaked, or maybe they are. They were produced by Colossal, a company one of whose big goals is to bring back the woolly mammoth. And so they took mouse genes, mouse variants that were known to produce phenotypes, variations in mouse bodies that were similar to what we see in woolly mammoths. They really didn't use woolly mammoth genes very much. They modified 7 genes, 5 of them were mouse genes, 2 of them were woolly mammoth genes. And they've got this cute little mouse. They are cute. They're golden colored and they got nice long hair. We don't know how well they do with cold because nobody's tried it yet. We don't know whether anything that happened there would happen in elephants, but it certainly makes sense to work with mice that are small and easy and have a 20-day gestation period as opposed to Asian elephants, which are big and we don't understand well, and have a 22-month gestation period. Think about being pregnant for 22 months or waiting 22 months to see if your experiment worked. So this is a step toward de-extinction. De-extinction, people try to argue there would be wonderful things from de-extinction, like reversing climate change by having woolly mammoths improve the permafrost, preserve the permafrost. There is an argument there, and I think there's probably at least a 1 or 2% chance that it's accurate if you could get 1 million mammoths roaming the tundra. But we will probably do this, not for that, but because it would be cool. Coolness, I mean, when we weigh pluses and minuses, we usually don't count cool on the plus side. Much of what I do in life, I do because I think it's going to be cool. Like all foreign travel, I can kind of say, well, it's educational. Yeah, right, you really want to go because it's cool. You want to see these things that you've never seen before. Much of what we do is cool. How many of you would be willing to pay, say, for you students later when you've got money, $100 to go to a safari park and see mammoths, saber-toothed cats, giant ground sloths, and dire wolves? Raise your hand if you pay that. I don't believe those of you who don't have your hands raised. I certainly would. But de-extinction is one use, but I think we'll see other things. Any of you rose gardeners, growers? Okay. You ever seen a blue rose? No. No. People have tried for hundreds of years to breed blue roses. It shouldn't be hard to take a white rose and add blue pigment creating genes. Green poodles, why not? It's silly. There's no good reason for it. We will do it. So that's another area of genetic stuff that we will do that some people will be happy about, some people won't. Things like cloning pets. How many of you would clone a pet, assuming you had $50,000 you didn't otherwise need? You could get your pets cloned, and people do them and say they're very happy about them. How many of you, well, I won't put it that way. Let's say there's a couple who have one deeply beloved, incredibly brilliant teenage son. They're rich as Croesus, they are old, they can't have any more kids, and their son dies of typhoid fever in Florence, Italy in 1882. And their names are Leland and Jane Stanford. And his name is Leland Stanford Jr. I am a graduate of and work at the world's only junior university. It's Leland Stanford Junior University. Well, if they could have cloned little Leland, Andrea and I might never have worked, there might not never have been a Stanford University for us to work at. And her alma mater of Berkeley might have won more big games. So, yeah, that's a fair point. I'll give you that. So there are all these things on the edge that are concerning. Now, Act 2. It's harder than we expected. And this is true in two ways. And I want to take you back, for those of you who can go back that far, which is certainly not all of you, to the year 2000. The Human Genome Sequence draft form was announced. Human embryonic stem cells had just been discovered. And functional magnetic resonance imaging was telling us amazing things about how the brain works. And reasonable people, not just newspaper headlines, were saying, in 20 years, we're going to cure everything. These new tools are amazing. They're going to teach us how to do basically everything. Real people, including, say, Francis Collins, were talking about the huge medical revolution within 20 years. It hasn't happened. Good things have happened. It was worth it. Good things have happened in all those spaces. But they haven't been revolutionary. Why? Because biology is weird. And it's more complicated than we thought. So take gene therapy. The idea goes back at least to the 70s with a guy from UC San Diego named Friedman. The first illegal attempt was made at UCLA, well, made by UCLA prof in Israel and Italy in 1981. Then it was tried at Penn in 1990. It now works, kind of. It works for people where it's a blood disease, so you can take the blood-forming stem cells out and manipulate them in the lab and make sure you've got the right things. You can put it back in, and sometimes it works. You have to usually wipe out their current blood-forming stem cells, which requires nasty chemo or radiation therapy and has a non-trivial risk of death and a certainty of discomfort. But for other diseases, if you need to, say, change somebody's heart, how do you get those DNA to go to the heart, and only to the heart? You don't want active heart DNA in the wrong place. You want to make sure that it stays what you've changed it into and doesn't change back to. You want to make sure there are no pluripotent cells in there still that might turn into a tumor. How do you do all that? And then how do you deliver it? We mainly deliver them through viruses, although they're a variety of different ways. But there's worry if you use the adeno-associated virus for one try, maybe the person's going to develop antibodies to that and you can't do it again. But maybe you'll get a better form, a better CRISPR technique, but if you can't use the virus again. So as a result, we can do gene therapy. It's been approved for about 20 different indications in the US and Europe. And it's almost never happening. It's been harder than we expected. The same is true of fMRI, which is amazingly good at telling you what parts of the brain are activated in 80% of people when they do or see something. But the problem is we aren't usually that interested in 80% of the people. We're interested in you, or you, or you. And 80% is not good enough to know whether you're in pain or whether you're lying, or all these other things. And similarly with genetics, it's turning out to be more complicated than we thought through things like epigenetics and things like little bits of DNA that are floating around in the cell doing things we don't understand, and stuff that's going on wonderfully here in condensates, these bits of stuff inside cells that are basically organelles without membranes, and we don't understand them yet. You'll fix that, right? Okay, so it's harder than we thought. I do not think it is infinitely hard. Well, maybe ultimately it is infinitely hard, but I think getting good results will not be infinitely hard. But it's going to take longer than we thought. But it's not just the science. It's also the society. People get nervous about this kind of thing. Sometimes they get nervous for reasons they call ethical. There are ethical concerns that I think are really quite serious. Some of this stuff is genuinely dangerous, like mere life. Maybe it won't be a danger, but it could be. We should worry about that. Some of it, like super babies and a genetically created and maintained caste system, I think is always going to be science fiction, but it's not completely impossible. That's a concern. Other things remind me of the days when, in the early days of heart transplants, when people were freaked out that a man might get a woman's heart and turn into a woman, or a white man with a black man's heart would change. This was real, especially since much of the early work was done in South Africa. So there is stuff that doesn't make any logical sense, but it doesn't matter at some level whether it makes logical sense or not. If it freaks people out, then that means getting political support, avoiding protesters, getting people to come into your lab in an area where their parents say, "You're doing what?" All that gets harder. So they're the social side, and then, all these drugs, they need to make money. One of the saddest stories I know about involves actually SCID, not the Omenn SCID, but SCID ADA, one of the more common, one of the less rare SCIDs. There was a company that, using money from the Californian Institute for Regenerative Medicine, came up with a really effective gene therapy cure for SCID ADA. Worked for 48 out of 50 kids with no problems, except they realized there was no way in hell they could make money off of it. So they gave it up. They couldn't figure out a way to charge enough money and get repaid enough money to make it sensible to their investors and the venture capitalists behind them to treat 100 people a year. There is a gene therapy for sickle cell. Sickle cell, it turns out, most people can live with sickle cell, and so if you're not that bad off, the immediate problems of gene therapy are disconcerting, and all the uncertainties. These companies lost, it was CRISPR Therapeutics and Vertex, lost $560 million last year on their sickle cell drug. They sold it to 5 people for $3 million each. But that doesn't make up stuff. So society isn't ready for some of these things. The problems, it's more complicated than we thought, isn't just on the science side, it's on the social side. So part 3, Frankenstein. Do not think about Boris Karloff. Think about Mary Wollstonecraft Shelley, the 18-year-old who amazingly wrote this book in a summer at Lake Geneva with Lord Byron and her husband, Percy Bysshe Shelley. I think they weren't married at that point, but they did get married. It's an amazing book. People think of it in the movie, very different from the movie. People think of it as the story is the story of Prometheus. There are things that man should not do. We are presumptuous. We go too far, and if we do, we get knocked down. I have written a piece, I was not the first person by any means to suggest this. I'm not a literary theorist. But I argue with other people, it's not about that at all. Frankenstein is about parental duties. The medical student, Victor Frankenstein, took one look at his monster when it woke up and ran away screaming. He never cared for it, he never trained it, he never helped it. Turned out it's the most eloquent and intelligent character in the entire book. Very different from the movies. And he neglected it entirely. This is sort of, I'm not a psychiatrist licensed anywhere, but Shelley's mother, Mary Wollstonecraft, an early feminist, died after giving birth to her. Her father quickly remarried a woman who hated her, and they sent her off to live someplace else. Parental betrayal, I think, makes a lot of sense for Frankenstein. She did a second edition in 1831, which was a little more conservative. But what does this have to do with bioscience? These things we make, they are in some sense our children. We need to pay attention to them. We need to care about them. We need to watch them. We need to monitor them. We need to make sure that they go in good ways and not bad ways. And unlike our children, we need to kill them off if things look too bad. We need to be attentive. We need to be attentive not just as scientists, but as a society. And part of that being attentive as a society is for scientists to be attentive to the society. Again, a few of you probably remember Dolly the sheep. Dolly the sheep prompted hysteria. One cloned sheep born, not the first cloned mammal, not the first cloned vertebrate, but the first clone from cells from a living born mammal. And suddenly the headlines are full of armies of cloned warrior slaves. 50 countries and 20 states passed laws banning human cloning, which had never been done and still hasn't been done as far as we know. People didn't expect it. They were blindsided. Science needs to not blindside people. If you've got something really startling, and I know this runs against issues of confidentiality and the race to win, the race to Stockholm, and all the other reasons scientists don't want to publicize their work too much. But if it's something you can predict is going to be controversial, prepare a pathway so the public knows why you're doing it, why it makes sense, is not shocked, does not react hysterically. Part of that, I think, was Hu Zhonghui's problem. Hu Zhonghui had many problems, but part of it was nobody expected this. It came out of the blue. So we need to scan the horizon to see what's coming. We need to inform people about what we're doing and have a conversation, not try to lecture to them about why this is a good thing and why we're right, but talk to them and listen to them about what worries them and try to explain that's why they should or shouldn't be worried. And sometimes, change your research topic or change your research in some ways. Ultimately, science cannot live divorced from its society. We have to be part of our society. We can't break the laws. We need funding. We need approbation and approval. We are not able to divorce ourselves entirely from our society, and it ultimately makes the rules. We have to try to have a conversation with it that allows us to do and to convince them that we're doing good work. We also need to monitor stuff. And this reminds me of university things. It turns out it's surprisingly easy, I am told, I've never done it myself, to raise $100 million to build a new building. As long as you can put the person's name on it and other things. It's really hard to raise $20 million to endow the janitorial fund, or the supplies, or the lights. This is part of the whole indirect cost thing that's going on now. Maintenance is not something our culture values. Maintenance is crucial. And in this context, maintenance means don't just have the FDA approve it and go away. Follow it up, see what's happening, watch what actually occurs, and if necessary, make changes. So we've got miracles and wonders. We've got it's harder than it looked. We've got Frankenstein. I hope in this time I have convinced you that this is an amazing time to be alive, especially if you're interested in biology at all. We know approximately infinitely more than we did 30 years ago. 30 years from now, they will look back and say, those fools thought they knew something. They knew nothing. And both things are right. It is an amazing time to be alive with great possibilities, but also dangers, some of them real, some of them social, some of them ethical, but things that we have to take into account. We as members of society, we as voters, you as scientists. My undergraduate degree from Stanford is in science, but it's in political science. Most of the science I work on is very political, so it's not entirely wrong. Pay attention to this stuff. It's important. It's also really interesting and fun to learn about. So when you do your science, think about explaining it to your cousin who doesn't know anything, or your aunt or your uncle, and how you would explain it to them and what reactions you're going to get. And think about how that could affect people you know from your high school classes and so on. We all live in bubbles. We need to be aware of those bubbles and not be like David Vedder, the boy in the bubble. We need to get outside of those bubbles in order for science, and I believe society, to progress in ways that reach our goals of a better, cleaner, more humane world with less suffering. Thank you very much. So I'm hoping I left a little time for questions. I left 10 minutes. Hey, I never do that.
Perfect.
I always say I'm going to leave 10 minutes. I generally leave 2. So any questions? Or comments, or don't throw fruit, but otherwise.
Hi, thank you for such a great talk. The last few years have taught us, especially the COVID times, that it's difficult to convince people of science, or it's difficult for people to understand science. It's not their fault, it's our community's fault. And that was just factual.
There's plenty of fault to go around.
Yes, but now we're talking about science that also involves ethics, right?
Yeah.
So if it's difficult for us to convince people of just facts, how are we trying to get people on board when facts and ethics and everything else is involved? So what should we do?
So 2 things. And first, you reminded me, I love working without notes, but one of the disadvantages is sometimes you forget something you would plan to say, which said one of the things we should not do is arbitrarily fire a bunch of scientists, cut budgets, slash indirect costs, be cruel in every possible way to all of your boss's enemies. And that's part of what's going on. So, and the last 5 years have clearly seen a huge decline in public support for reason and rationality and thinking things through. It's a wish fulfillment. Wish fulfillment as a way of life has become much more attractive to people. What I think you can do is try to explain what you're doing and why. Listen to questions, listen to concerns, respond to the concerns when you can. You are not going to convince everybody. There is nothing that all 8 billion people in this world will agree to, or all 330 million people in this country. But you do need a majority of people who either support or don't oppose to get things moving forward. And how do we resolve it when one group says yes and one group says no? Well, Churchill said, democracy is the worst form of government, except for all the others that have from time to time been tried by the mind of man. In democratic countries, which we still are, for now, I think that ultimately the political process makes those decisions, and you try to influence the political process as you can. But people hate being preached to. I say that even though I kind of preach myself. But so you don't want to say, okay, you ignorant yokels, here's what science says, this is right, and you're dumb. That doesn't convince a lot of people, other than it convinces them that you're a jerk. So having a good conversation with people, recognizing that you'll never get perfect agreement, either on the facts or on the ethics, but respecting their ethics. I'm a pro-choice person, but there are aspects of abortion that I find deeply troubling. And I have pro-life friends whose views I respect, particularly those who are consistent. So they're in favor of, they're against abortion, but they're also against capital punishment, for example. But respect ethical views that you don't share, but say, look, I don't share them. And hope that you can get a majority. Oh, you got a guy right there. You were a good audience member. You were nodding at all the right times. You guys think that we never pay any attention to you? But, you know, I didn't see many of you shopping on the internet, so that was good.
As genetic tools have become more available, proliferating, and the skills to use them have become more widely known.
Yep.
It's become possible for people to manipulate their own genomes.
Yes, kind
What
of.
right do they have to
So that's a really interesting question. There's a biohacker who used to be Josiah Sainer, who is now Joe Zaner, who went on TV, live streamed himself, injecting himself with CRISPR, and dared the FDA to come and get him. Because the FDA's view is this is illegal. This is doing an unauthorized introduction of an unapproved drug into interstate commerce. FDA didn't do anything, and ultimately it said, we got other things to worry about. If he wants to do that, it's illegal, but we're not going to worry about it. However, I think the bigger issue is with the rise of genetic tools, on eBay, you can buy used copies of a whole bunch of stuff you would need to do things. There are a lot of biohackers out there who are genuinely science enthusiasts. It's like the chemistry kit you had as a kid, only it's better. They're going to try to do some cool stuff. Somebody tried to make glow-in-the-dark beer. It didn't work very well, but there's some residue at the bottom that glowed green. Probably not very attractive. But I worry that they're going to try to do something they think is going to be good, and it's going to turn into a plague or a pest. And then you have to worry about the people who want there to be a plague or a pest. The increased accessibility of CRISPR and similar tools and the knowledge and the machines to do them could make biowarfare against humans or against our crops or our animals a lot easier. Solving that problem is really hard. The good news is it takes more than a pathogen to have a good bioweapon, but being able to make a good pathogen. We know the code for smallpox. We know the code and have examples of horsepox, mousepox, monkeypox, and other poxes. Could somebody take monkeypox and turn it into smallpox? I don't know why not. And I'm vaccinated against smallpox, but most of you guys are not. You're too young. So yeah, it's a worry. I think I've still got the scar up here.
Maybe one more question if there's one. Anyone like to take the last question? I guess
Take the last question.
I'll take why not? I think the thing that gets the most headlines, probably for good reason, is CRISPR babies and germline editing, and some of the books on this talk about the future of humanity. If you take a person from 1,000 years ago and you brush their teeth and clean them up, you wouldn't notice, but maybe if you take somebody from 1,000 years in the future, you'll notice. Do you think that we're at a turning point in the evolution of humanity in that way?
So humanity is evolving, has been evolving, continues to evolve. Everybody in this room has multiple copies, I'm predicting, of a gene that makes a protein called amylase, which helps you break down protein. It is starch, turns starch into sugars. It's really useful in agricultural societies. It seems to have had two big spikes in its introduction into humans, a small one about 40,000 years ago and a big one about 6,000 years ago, about the time agriculture got started. So we have evolved to have more copies of something that helps us digest starches because we started eating more starches. And the people who had multiple copies did better, and their kids who inherited them did better. My cousin Mike had type 1 diabetes, juvenile diabetes, from the age of 8. 103 years ago now, two interfering Canadians, those damn Canadians, figured out how to keep all those people from dying before they reached puberty. Juvenile onset diabetes, before insulin was available, it was deadly, and it was a juvenile disease. They didn't reproduce. They didn't live long enough to reproduce. Type 1 diabetes is not heavily genetic, but there are genetic variations, mainly in your immune system, in the HLA system, that greatly increase your risk of getting type 1 diabetes. My prediction, I'd love to see somebody try and actually do this, is that there are a lot more of those negative alleles around today. Because my cousin Mike, who should have died when he was 8, died at 69 with 16 grandchildren. He's passing that gene on. Now, some would say, medicine is making us all weaklings and turning us into a weak, degenerate race. Well, only if we get rid of medicine. I think that we are evolving, we will continue to evolve. The great fear is enhancement and super babies and caste systems. And I just can't get myself worried about it because brains make everything else I know look simple. We could, change your hair color, right, but you can do that with dye. Change your eye color, we can do that with contact lenses. I don't care if somebody turns their baby's eye color from brown to green or selects for light-colored eyes rather than dark-colored eyes. It doesn't matter. If we could make them into great athletes, or great mathematicians, or great chess players, or great law professors, or even great scientists for that matter, that might be a different story. But we don't have a clue how to do that, and I don't think we will. I know we won't during my lifetime. I don't think we will during any of your lifetimes. We know a whole bunch of genes that are heavily, heavily involved with major changes in intelligence. They all make intelligence a lot lower in certain variations. Even the most optimistic people, if that's the right word, believe that they've identified genetic variations that account for 1 or 2 IQ points, which is below trivial. So, yes, it gets all the attention. I don't think it deserves it, even though I wrote a 300-page book about it.
All right, before we let everybody go, we have a little memento of the time here at Princeton. So this is it says presented to Hank Greely, Gilbert S. Omenn, MD, PhD, 61, and Martha A. Darling, 70 lecture in Ethics and Policy in Bioengineering 2025. We can ship that to you, so if you're worried about it.
Shipping would be good.
Yeah, sure. We'll do that. I know you're internationally traveling. Maybe you want to get a picture right while we're up here?
Well, how about getting Gil and Martha up?
Oh, of course. Yeah, we can. Yeah, that's perfect. So let's thank.
This is heavy. I could do serious damage. This is kind of sharp.
That's why the best thing is
There's
the
a
shipping.
reception out here in the hall. We want to invite everybody to come. We'll have a couple little remarks there, and thank you all for coming.